Hello everyone, thank you for having me here. My name is Casey Donner. I currently reside in Southern California. I took Dr. Elaine Ingham's Soil Food Web course online, and I'll briefly tell you about that and then go into what I learned about composting from her course. And I may ask you a few questions along the way, so feel free to participate. Who is Dr. Ingham, and what is going on in her compost pile? Dr. Elaine Ingham is a soil biologist who is widely recognized for advancing knowledge about the soil food web for over 4 decades. She's passionate about empowering ordinary people to improve the health of soils in their communities. Like all of us composters, Dr. Elaine is mimicking a natural phenomenon called decomposition to create an ideal soil amendment, using waste products that would otherwise fill a landfill and emit more potent greenhouse gases into the environment. But she also monitors the predators and prey living in the soil by counting them, and then balances the soil food web organisms when they have been disturbed, such as by novel chemicals. So her compost looks very much like the O horizon of the soil of whichever plant she is trying to grow, and with all of the predators and prey in balance, except her organisms are indigenous to wherever the compost pile was built, and that is what is meant by biocomplete compost. Dr. Elaine and the Soil Food Web School instructors offer content online showing the results of many of their projects and case studies using the Soil Food Web method, which really is just following some basic principles about composting and then checking your work. Why is it important for communities to make their own local compost? It's important because if you're buying it from a big box store or a high-end fancy composting facility 2 states away, probably only half of the microorganisms are going to make it when they get to your environment. The composting principles in order. Calibrate thermometers. It'll be best if you can buy a thermometer that actually hits 212 degrees Fahrenheit. As you can see, mine doesn't. Dip the tip in a bath of boiling water and an ice bath to ensure accuracy of measurement, and use your adjustment screw when needed. For those thermometers that do not have a calibration screw, it's useful to know how off they are and then label the difference on the backside. Piles should have enough mass to hold heat. Dr. Lane builds small piles on pallets to increase airflow from the bottom as hot air rises from the top. But for windrow piles, she doesn't yet recommend active aeration because she hasn't seen examples of it evenly diffusing air through the pile. I'll give a side note here. I think this can be remedied, and I think this would actually be the ideal method for community composters. Dr. Elaine also recommends using waterproof tarps to maintain moisture, but to tent the tarp to maintain aerobic conditions under the tarp. Shelters are better, but not everybody has that option. All green feedstocks must be fresh or preserved. Options include freezing, dehydrating, or drying. No green feedstocks should have set out for more than 3 days because putrid odors rising from rotting feedstocks are nutrients off-gassing. However, browns, as long as they are not kept waterlogged, they do well sitting out because they can just capture fungi and break down a little bit. Defrost frozen feedstocks and rehydrate dried feedstocks and aged wood chips for about 8 hours before you build your pile. This helps to maintain the ideal moisture during the composting process. Soil microbes live within the thin films of water in soil and on plant surfaces, so if this step is skipped, piles can sometimes dry out during the hot composting process and temperatures may not remain high. Water quality matters. Chlorine and chloramines in tap water protect us from harmful pathogens, but they can also negatively impact the beneficial organisms in a compost pile. The best option is to use humic acid in a hose end sprayer. Chlorine filters are another option, but cannot filter out chloramines. You could run humic acid through a fertigation system, but if you buy it, some humic acid is mined, probably all of it, and Soil Food Web School seems to recommend more sustainable approaches to get the desired result. So where do you get humic acid if you haven't built your first pile? Well, it works without it, just not as well, so you'll eventually get there, or you could get some compost from a friend. Aim for a diversity of feedstocks to promote a diversity of microorganisms. The more diverse the gene pool, the more likely the community is to survive. Diversity also promotes abundance and health in nature. I use a variety of manures to encourage a wider diversity of microbes, but always assume manures contain human pathogens. Anyone who can guess what manures these are? Feedstocks with strong aromas may have antimicrobial properties that may affect your pile's temperature. It's best to let aromatic piles age. Pine wood chips, for example, could age for 6 months with some aeration before using as a starting material. Some amendments, microbial foods, may contain preservatives, which are antimicrobial. Follow dilution instructions before adding fish emulsion to the pile. I like to use cracked corn if I'm struggling to keep my piles hot, but I rinse it first because I imagine it probably has some kind of preservatives on it. 10% of your pile may consist of questionable materials such as greasy paper, roasted chicken carcass, Q-tips, cooked food, leftover salad, dead rats, but plastic is not one of them. And place potential pest attractors in the hot middle when you build your pile. An easy way to think about the carbon to nitrogen ratios of your feedstocks is that plants dedicate energy to specific parts depending on the season. During the flowering stage, the plant is sending its energy to the flower. During the seeding stage, much energy is being dedicated to the seed. For the biennials and perennials, during winter, the energy is stored in the root. Why do we count coffee grounds as greens if they are brown? Because they were harvested when they were ripe. Feedstocks that are harvested while fresh and dried for storage count as greens. Is grass still high in nitrogen after it's been dried? It's definitely still high in nitrogen, but how high is it? I actually don't know. I imagine that as it dries out, it loses some of its nitrogen. Feedstock should be about 1 cubic inch. Using a lot of sawdust could reduce oxygen flow in your pile, but using a bunch of large branches, well, they won't compost very quickly, and they might even dry your pile out.
Ready, set.
And yes, this includes whole fruits such as oranges, as well as manure balls, which must be crushed. Otherwise, the center of these often become anaerobic, which we avoid. Large scale if you're using front loaders, while the driver's getting a new scoop, people on the ground can be crunching up and breaking up balled up materials. For those materials that mat easily together, such as grass clippings or juice pulp, mix those in very well to avoid anaerobic conditions. We use 5-gallon buckets as measuring cups, which makes it easier to get the recipe right. The base recipe is 60% browns, 30% greens, 10% high nitrogen materials. Here I'm building a 50 bucket pile with 30 buckets of browns, 15 buckets of greens, and 5 buckets of high nitrogen materials. Now this is the basic recipe. it is necessary to test your inputs and record your results. There are many factors that affect the heat generated by your pile. For example, the CN ratio of coniferous trees is 500 to 1, which is higher than that of deciduous trees, 300 to 1. And for those of you living in snow country, it's likely that 20% of your pile will have to consist of the high nitrogen materials in order to maintain heat. So what are high nitrogen materials? Dr. Elaine calls them party foods. They'll have a CN ratio of 5 to 1 to 10 to 1 generally. They include meat, meat broth, blood meal, bone meal, if it has marrow in it, fish hydrolysate or emulsion, manures of chickens, cows, pigs, horses. The CN ratio of those is highly dependent on their diets, and it also includes green legumes if they are cut while they're green and if they have red nodules on their roots. Here's a little quiz for you. Whole eggs, are they green, brown, or high nitrogen? Sheep's wool, is it a green, is it a brown, or is it a high nitrogen material? The squeeze test. When you're mixing your feedstocks, building your pile, the starting material should be at 50% free water. We're not worried about gravimetric water here. We want to measure the amount of water that will drip out of your hand when you squeeze it. So put some mixed feedstocks in your hand. Squeeze them in your fist as hard as you can. 1 to 2 drops of liquid should squeeze out of that fistful of material. If you get no liquid drops coming out of the material, it's not wet enough. After you open your hand, it holds its shape. It's probably at 40% moisture content. If after you open your hand, the material falls apart, it's probably 30% less moisture content. And if it has more than 1 to 2 drops, then it's too wet. And add some dry ingredients. Do at least 3 squeeze tests and take the average of those anytime you mix ingredients to build a pile. Immediately after you are finished building your compost pile, power rinse the receptacles that the starting materials were soaked in to reduce the chance of biofilms developing and harboring pathogens or pests. In larger facilities, this may be more of an issue. Some points on thermometer handling. Never hold or grab your thermometer by the head. Grab it 6 inches under the shaft. Protect your thermometer with a sheath. Mine is PVC. The tip is where the temperature is read, so make sure your point is in the hot center. And if you can, use multiple thermometers to gauge the hot middle versus the cool edges. Monitor your pile temperature several times a day. It needs to be hot enough, long enough. But in order to kill pathogens, pests, and weed seeds, every part of the pile is heat treated at 131 to 149 degrees Fahrenheit for 72 hours, or 150 to 159 degrees for 48 hours, or 160 to 165 degrees for 24 hours. After you build your pile, temperatures begin to increase and the time starts ticking when the pile hits 131. If the pile hits 150, the second timer starts, and if it hits 160, a third timer starts. After 24 hours at 160 degrees or above, the pile gets turned. If the pile goes above 165 degrees, you're starting to risk the chance of going anaerobic because you have so much bacteria in this pile. They're taking up oxygen faster than oxygen can diffuse back into the pile. Plus, you start damaging your fungal populations at 150 degrees Fahrenheit. To keep the pile from getting too hot or anaerobic, add chimneys to introduce airflow. We don't leave PVC pipes inside the pile because they don't aerate as well as the chimney alone. The theory of turning a pile. When the pile heats up, it's really only about a third of the pile in the center that is sufficiently heat treated. And so we divide that pile into three: the top bowl, the center, and the bottom bowl. After the first heat, the pile is aerated as we turn. The top is removed and set aside. The center core becomes the new bottom. The original top becomes the new center core, and the bottom goes on top. After 3 heat treatments, all parts of the pile have been hot enough, long enough. Your pile should now be free of pests, pathogens, and weed seeds. Hold on a minute. Organic standards require you to turn your pile 5 times in a period of 15 days, during which the pile should be above 131 degrees. Are you telling me that the Soil Food Web School can do this faster? That is the claim. While the Soil Food Web School often must work under organic standards, when they do not have to, their research suggests the more the compost is turned, the fungi are disturbed, which can make it difficult to produce compost that matches the soil profile of the horizon of trees, shrubs, wheat, corn, and any plant requiring some fungal dominance. During the turning process, Soil Food Web School instructs us not to add any new materials to the pile, as we would be reintroducing potential pathogens after just getting rid of them through heat treating part of the pile. The exception being when your pile is just barely hovering above the baseline temperature early in the process, blood meal may be added to the pile to bump up the heat, or materials could be added to the hot center of the pile only, which is very difficult to do unless you turn piles like this, which no sane person would do unless you were to inoculate many larger piles with the amazing biology extracted from one of your BioComplete piles. Think about it. Use your senses to judge quality during the turning process and after the pile is cured. What does your compost pile smell like? Well, when I first build mine, it smells like horse manure, and so do I. But after 24 hours, if I smell the ammonia smell, I know I'm losing nitrogen. And if I smell the rotten egg smell, I know I'm losing phosphorus. Other smells include the rotting flesh smell called putrescine, the vomit smell called valeric acid, the rotten milk smell called butyric acid. It's under anaerobic conditions or reduced oxygen conditions that these smells will take place. If you smell a slight musty odor, it could mean that your compost material is waterlogged and it's on its way to reduced oxygen conditions. If you smell the gristlefolvin or the petrichor, geosmin smell, the smell after a rain, that might be indicative of actinobacteria, which they're okay, but they might not be desirable in composts that are going to be used for trees or shrubs. And lastly, the smell that we really want is the smell of an old-growth forest soil or the smell of mushrooms. So use your eyes. What do you see? Perhaps you see some pests. The larval stages of flies, for example, need a lot of moisture, and so it indicates that your piles were greater than 60% moisture. Also, having pests is an indicator that your pile may not have been hot enough, long enough at all parts of the stage, or else it would have heat treated for those pests. Do you see fuzzy fungal growth, which is not the greatest sign, or do you see strand or cord-like fungal growth, which is a better sign? Do you see mushrooms growing in your compost? The good thing is, at least your pile doesn't have any chemicals in it that are inhibiting fungal growth. The bad thing is, you actually don't want any one fungi to dominate. You want them all to be suppressing each other microscopically so that not one can dominate and actually grow into a full mushroom. Do you see something that looks like white ashy material all over your pile? Actinobacteria is indicative of lower oxygen conditions than what is optimal. So if your pile's looking really ashy, you might want to water it less or turn it more or find a way to get more air into your pile. Especially if there's an actinobacteria layer and then there's a dark black layer of compost under that. Black colored composts are associated with anaerobic nitrous oxide. Soil Food Web School recommends that compost and compost-derived liquids be the same color as 70% cocoa brown chocolate. When you mix compost in water and stir it up, some of the contents of the compost are dissolved into the water. If after a few minutes of settling, the water is tinted a color, then that's a good sign. If the water is clear, that's a really bad sign. If the water is tilted 70% cocoa brown, that's indicative of humic acid. If it's a honey color, that's indicative of fulvic acids, which are good but not as good as humic acids. When checking for texture, hold compost in the cup of your hand and just shake it a little bit. If the compost stays in your hand, that's a good sign. If it falls right off, that's not a great sign. Bacterial glues are holding soil aggregates together and keeping it moist and creating structure. So when you look at the compost, you should see little airways and passageways. You should see little balls of soil connected together instead of like a floury consistency. You want a balled up consistency, little tiny soil aggregates. And then compost can also be evaluated microscopically by counting and measuring the organisms in your compost samples. I use microscopy to determine the fungal to bacterial ratio of the compost I've made, to potentially alter it so that I can feed specific plants with the organisms that are going to help them get the nutrients they want from the sand, silt, and clay particles. Thank you for your time. Thank you for letting me share. If you have any questions, feel free to reach out to me, and I'm hoping that I provided you with something fun or valuable today. Thank you.